the बैंक अकाउंट प्रॉपर्ली मेंटेन होते रहे, जो भी अनस्पेंड अमाउंट है टाइमली अगर वापस चला जाएगा, I think we should start. The students which are joining will let them join. But today is the last day, so we'll be having certain formalities in the end also, sir. We'll be having with us principal of the college and HOD too. So I think we should start.
Okay. I'll start.
Yes, sir. I welcome you, sir, on this third and conclusive day of the academic web series or lecture series. And students, I welcome you too. You have right till now you have been through all the nuances of NMR. That what NMR is. Now, sir is going to tell us something further about NMR. And as promised, we will be having a graded assignment that will be uploaded at 8 p.m. Today, and it will be closed by 10 p.m. The assignment is already being given by sir. Sir has given us the questions. So I'll be uploading that assignment which sir has given us. And the window for that is 8 to 10 p.m. After 10 p.m., I will be closing the assignment for responses and will not be taking the assignment for responses. This assignment will be uploaded on Google Classrooms, hosted for MSc first semester and MSc final semester. That means MSc third semester. So I hope and I request you to please go through the assignment at 8 p.m. sharp and revert it back by 10 p.m. Thank you. And now I request Dr. S. Ragothama to please go ahead. Thank you, sir.
Okay. Thank you, Dr. Kalpana. Okay. So good morning, everyone. We come to the final session of this web series. So last 2 days I covered, we learned about some basics of NMR and useful parameters in them. And now today I'll be telling a little more about two-dimensional NMR and eventually how all these things are helpful for getting structures or the characterizations of chemical molecules and all. Okay. Today mainly I concentrate on introduction to the 2D NMR. Okay. To start with, as I said, we learned 1D NMR over the last 2 days. And now in the top picture shows the 1D NMR, simplest 1D NMR pulse sequence. There are not only one pulsar, there could be many pulsars. The basic experiment is as soon as you put your sample, it can be divided into 2 time intervals called the preparation time and the detection time. So as soon as you put your sample in the magnet, you have to wait for a system to get into equilibrium. That's called the preparation time. And then you send a pulse and acquire the acquisition time is called the detection time. Now you get a time domain signal, you Fourier analyze this, Fourier transform it. That's why it's called FTNMR technique. So once you Fourier analyze this, the time domain is converted to frequency domain, which you're all familiar with, and we interpret that data. Now, when we move on to the 2D, the NMR gets spread over 2 dimensions. Both would be the frequency axis. And now there are 4 time intervals. Now the preparation and detection remains on either end, but there will be 2 more time intervals in between. Now, as I said to begin with, system has to equilibrate. We call that preparation period. And then instead of detecting, we will not detect it, but we will allow to evolve it in the time. That's why it's called evolution time, but there is no detection here. And eventually, once the evolution is done, we have what is known as a mixing period. So this is what determines what kind of experiments we do, whether we do a correlated experiment, a COSY, a NOESY, a TOCSY, different kinds of experiments we have. So that decides what kind of mixing pulses we should use. It could be a single pulse, it could be no pulse, it could be 2 pulses with some interval. There are various versions here. So the information we want, that depends on how we mix them. And eventually you detect it. So there are 2 time intervals one now. Because it evolves in 2 times, it's called 2-dimensional NMR. Now you have to do a double Fourier transformation because there are 2 dimensions. Now that's why it's called a 2D spectrum. Now what is 3D spectrum? It's further extension of this 2D spectrum. Again, you can even think of it as a combination of two 2D spectrum. Don't detect, don't prepare, add them. So you start with, again, preparation period, evolve in the time T1, mix it in particular way, don't detect it, evolve it in, again, in some other fashion, mix it, and then eventually detect it. So there are 3 term domains, and then you have to do triple Fourier transformation and eventually get the 3D spectrum. Like that, there is a 4D, there's a 5D, and there can be ND. But soon you lose your signal, so you cannot do more than 5 or 60 practically. But people are tempted 60, even up to 80, I think. So getting back, so these are all computer control, they're software control. So how to send this in simple 1D, this is what is known as the pulse program. And this is a kind of subroutine used to generate this pulse sequence. So ZD means 0, the memories, and this is the preparation period, it's called the D1, and then send a pulse called the P1, and the pulse is sent with certain phase, whether it has to come from x-axis or minus x-axis, y-axis, z-axis, and they are all coded in a particular number way. 0 means x, 2 means minus x, and then things like that. So you can send the pulse in the direction you want it. And once the pulse is over, you collect the data. Go means acquire the data. And again, there is a phase associated within the receiver, so let us not go into those details. So once this is done, you write it in your computer and come out. So this is the simplest pulse program we can think of. But when you do go to 2D and 3D, there will be a number of pages of pulse program. So let us not worry about it. Now, for this to activate, you have to choose a power levels of your pulse and pulse width of that, and how much time you should delay and all. So those things are given below. So eventually, when you do this, you get your 1D spectrum. So now, when we go to 2D spectrum, as I said, it could you won't get the one can get even the stack plot of 1D spectrum bunched to one above the other, but it's better to look at from the top and take the contours, and this is how it would look like. So now this is the spectrum of a lysozyme. 1D is quite complicated, so we need to resolve them, go to the second dimension. If you go to the second dimension, you see this is only a part of the second dimension. You see it's already even more complicated. So to resolve that, you pull it in the third dimension. This would be the 3D dimension. Even if it is not possible, you go to the fourth and fifth dimensions. Now, when you take slices of 2D, it would be nothing but a 1D spectrum. If you take slices of 3D, it's 2D spectrum. If you take slices of 4D, it would be a 3D spectrum. So it goes on like that, but we will stop at 2D spectrum for the current discussion. We will not go beyond that. Now, when you go to the 2D, it opens up a wide range of experiments for protein NMR or even other molecules, even slightly bigger molecules as well. Even in chemistry, they are very useful. Now, when you go from 2D to 3D, one can resolve many overlaps. It improves readability. There's a lot of advantage, a lot more information we get. You can even think it of as coupling two different 2Ds. If you project on one side, you get one kind of 2D. You project on the other side, you get another kind of 2D. So you can mix these 2D experiments by doing one 3D experiment. Now, as I said, if you project it, it's two different 2Ds. Now you have to do triple Fourier transformation to get the information from 3D. Now, as I said, we can keep on going. Now, as I said, because of overlap in 1Ds, to resolve them, we go to 2D. The 2D is also overlap. This is a simply contour representation where about 11, I think, NHS are overlapped in one particular slice. So to resolve that, you pull it according to the nitrogen dimensions. There are some separations. So now these 11 pieces get separated out, but still there are some overlaps. To do that, you now pull it according to the carbon dimensions. Now there is a possibility that each is individually separated out. So this is how you resolve going to the higher dimensions. now, most important thing here is when you do any experiments, multi-dimensional experiments, or even sometimes 1D experiments, you must be familiar with this coupling constants. Now, this is again the peptide sequence. I'm simply taking the example of peptide sequence, or even in organic chemist, carbon spectrum or nitrogen spectrum, you should know the coupling values. Normally, if it is carbon spectrum, is the CH coupling, which is important, which turns out to be 140 hertz, so that the timing between the pulses can be tuned to this. If time and frequency are related, one can pick and choose whatever they want. Now, if you want a nitrogen proton correlation, you should tune it to 90 hertz. Likewise, some other thing, the nitrogen C13 is about 15 hertz, nitrogen C alpha carbon is 11 hertz, alpha to CO is 55 hertz. So one should get familiar with all these coupling constants, because many of these multi-dimensional experiments, this information is needed. And as you keep on going to higher hand dimensions, experimental track keep increasing. And because as you, there are, time keeps on increasing, there are so many pulses, by the time you come to the end of the pulses, things would have already been relaxed. So that's the limitation of this multi-dimensional experiments. So now there are again methods to improve that, overcome the time factor. This is called NUS, non-uniform sampling. There are so many other techniques to overcome that. I'm not going to those details now. Okay. Now coming back to the 2D spectrum, here is the slide which explains the basic 2D experiments called the COSY. It's called the correlated spectroscopy, first given by Professor Jeener in a workshop. Probably I told you that, and he forgot about that. He simply introduced in a workshop. Luckily, it is documented somewhere. And then he didn't bother to take it up further. And then it's Professor Ernst, who then, as I told you yesterday, waited for a certain time over years of period, and then took his permission and went on to develop this technique. And it's so useful now. Everybody starts using it now. Now, what is a 2D? 2D is, now in this COSY experiment, it's called the correlation. It correlates between two protons. If they are spin-spin coupled, in 1D also, you can decouple one thing and see which is it coupled to. If there are hundreds of protons coupled to each other, you have to do 100 kinds of experiments. And here in 2D, in one big map, you get all these correlations. Now it starts with, and the COSY starts with the two pulse. I mean, the basic thing is a two pulse sequence. And then, as I said, this is the preparation period, and this is the evolution period. The time interval between the two 90-degree pulses is called the evolution period. And the last one is called the mixing. The mixing pulse is this 90-degree pulse, and eventually you detect it. Now you repeat it number of times. The only thing is you increase this evolution time. That is called the T1 time now. So they start with certain number called D0 and then multiples of D0. And you do some 256, 512 experiments depending on the time, depending on the resolutions you want. Now, if you do series of experiments and collect this data, your FIDs would look like that. To start with, the difference between these pulses is shown, and then you collect the FID. Now, you increase this time gap between these two pulses, and then when you collect your data, there is a phase change here, and then eventually collect it. And you keep on increasing and keep on collecting it. Now you have to Fourier transform. Now, if you Fourier transform only in this dimension, you get this figure. So now that would be the Fourier transformation of that. That would be the Fourier transformation of that. And you keep on Fourier transforming, you're literally generating another FID in the what is called the indirect dimension or the T1 dimension. Now you see another FID building up here. And now you Fourier transform this also, then, and all these Fourier transforms is much better understood. The study is much better understood if you learn a little bit mathematics. It's mainly trigonometry. So all these FIDs are either cosine or sine wise. And if you use this trigonometric function, then there is a they are simplified called by product operator formalism. but let us not go into those details. Essentially, during T1, it evolves in the chemical shift and also coupling, scalar couplings. So all those things are represented in these equations. And if you understand that, it becomes much simpler to generate these 2D or 3D spectra. So we are not going into those details. Now, pictorially, I'm going to represent how would a 2D look like. Now, this is yet another scheme. Instead of one pulse in the mixing time, we have two pulses with a constant time. And now this is called a NOESY spectrum, NOE information in the 2 dimensions. Now, if you do that, as I said, this would be the 2D of the whole view of the 2D FIDs. And now if you double Fourier transform that, it's a function of both T1 and T2. If you Fourier analyze it, you get 2 frequency domains on either axis. It's called, now it's converted to omega-1 and omega-2. And if there is only one kind of proton present, like water sample, what it gives only single peak, there is nothing else. So no point doing 2D for this. So now either you go in dimension or in this dimension, it comes at the center because both are same frequency units. And now if you take another 2 non-interacting species like water and acetone, again, you don't need 2D and it's also not coupled to each other. So you simply get 2 peaks on the diagonal. The frequency on this dimension or frequency dimension happens to be the same. But if you take a coupled spin system, like yesterday I was telling you, AX coupled spin system, then there is some information. Now, the 2 spins are represented on the diagonal. So the diagonal represents a 1D spectrum. Now you have this chemical shift, whether you read it from this side or this side, it will be the same. But now, because it is coupled to the second nuclei, the A is coupled to X nuclei, you see a peak on this dimension and the frequency modulated by that X frequency. So also when you look at the X chemical shift, it also gets modulated by the A frequency. So you see what is known as cross peaks. Now this is the diagonal, is the normal 1D spectrum, but if they are coupled to each other, you get what is known as off-diagonal peaks across this. So this is how a 2D spectrum, simplest 2D spectra would look like. Now you get this coupling information. Because there is only one coupling, there is only one kind of cross peaks there. Now normally 2D spectrum, as I said, is represented by contour plots. You look from the top and take this cross section. Now this is the diagonal peak, this is the cross peak. So that's how we analyze. Okay, let us look at some examples to have a better clarity. Now, this is a COSY spectrum, or called a correlated spectrum, and now they're correlated by the J coupling. So now, as I said, there are 2 pulses, and there is this preparation period, and there is this evolution period, and this is 90 degrees, the mixing pulse, and eventually you detect it. Now, I have taken the sample ethyl benzene, very regularly used. And if you do a 2D spectrum, and now you see this 1D spectrum goes on the diagonal. If you project this diagonal on this side, either on this side or on the other side, you get your normal 1D spectrum. And now you see this CH2 and the CH2 and the CH3s are coupled to each other. So there is a triplet and a quadret, and you can literally see those things across this diagonal. On either side of that, it appears. But they are not related to the benzene ring because this is 4 bonds away. So it simply gets isolated. And of course, among themselves, they are strongly coupled. Among themselves, they would be coupled. And now, again, there are kinds of COSY spectrum. When you do a COSY spectrum, there is what is known as gradient COSY spectrum, E-COSY spectrum, or super COSY spectrum, or double quantum filtered COSY spectrum, just to enhance the resolution or conserve time and all. But information-wise, they are all same. Only the quality of the information you get depends on what kind of experiments you do. Now, the main thing is to get this coupling information because, again, this is a simple molecule. There is only one connection here. And now you see there are fine resolution because This is known as double quantum filter, COSY. But if you want, we don't care about this resolution. We just want to see which is connected to what other nuclei. Then you can do a 5 minutes COSY. You'll get it in one shot, but you lose the resolution. Now that is the TMS. This is the CS3 and the CS2 that you see. You can do it in 5 minutes. Whereas the DQFC takes hours, at least 1 hour or 2 hours, get this resolution. So depending on the information you want, you can do that. Now continuing one more example, let us take L-propanol. It is CH3, CH2, CH2OH. You know CH3 comes at the one end of the spectrum. So let us see how to analyze this. So start with that one. Now the CH3 is coupled to the CH2. And now you see the coupling through this cross peak. And now this 2 is not only coupled to 1. So you can go backwards. It is also coupled to 3. Now the 2 is not only coupled to 1, it's also coupled to 3. Now when you come to 3, it is coupled to 2, as also it's coupled to 3 is coupled to 4 as well. So this is how you pick up, you correlate, which are all the J correlated here. Now, the other thing is in the COSY spectrum or the NOESY spectrum, it's symmetric spectrum. It's the information content is same on either side of the diagonal. That's why I put only on one side. So that's sufficient. Sometimes if you want more resolution, because there will be more resolution in this side, you can use this side as well. I can either use it this side or this side. One side of the diagonal is sufficient. Okay, going to the next spectrum, a little more complicated, is 2-butanol. Now again, there are 2 CH3s here. One is coupled next to the CH2, the other is next to the CH. Now because this CH3 is coupled to CH2, it would be a triplet. This CH3 is coupled to CH, it would be a doublet. So now you know the methyl, you can start from there. So let us start from the first methyl, and it's coupled to the CH2. Now this CH2 is coupled to the CH3. Now this CH3 you see is coupled to 2, 4, and 5. Now that's how you see 3 peaks connected to that. Now 3 is coupled to 4, 3 is coupled to 2, coupled to 4, and coupled to 5 as well. So as I said, in one shot, you get all these correlations in the total spectrum. That's the big advantage. And if you have even more complicated system, then all of them you can get in one shot. So now I again straight away gone to a peptide sample. This is a hexapeptide, cyclic hexapeptide. And now when you analyze your peptides, it becomes very important to assign all these peaks. So now there are certain patterns here I'll be telling you as we go along. And now I've simply picked up, now it's made up of phenylalanine, beta-glycine, leucine, proline, and phenylalanine, and I think valine there. Now I've simply taken 2 of these residues here just to explain this. Now this spectra is only a partial spectrum. I have omitted the NH peaks beyond 5 to 8 peaks. There are some more peaks. Because it gets crowded, I have removed that. Later on, I'll introduce that in some other case. Now I have taken only from this part. Now this connection is removed only from here. So alpha, beta, gamma. So they are all interconnected. Now how do we pull those connections? Normally this alpha comes around 4 to 5 ppm. So this valine alpha somewhere comes here. Now if you draw a perpendicular line here, it goes there, and that would be the alpha beta connection. Again, these alpha betas all have specific chemical shift values. Yesterday I was telling you in 1D spectrum. Now this is the valine alpha beta connection. Now from beta it has to go to the gamma CH3s, and there are 2 CH3s, it goes to that both CH3s. So that is how you can pull out the connectivities of this valine residue. So likewise, if you go to the leucine, there is one more step. So alpha to beta, beta to the gamma, gamma to the delta. So let us try to trace it. Again, the chemical shift position or specific positions. Now that is the alpha beta connections of the leucine, and that is the alpha connected to the beta. Now the beta is inequivalent beta. So this beta is not one peak, but there are 2 peaks. So there are this beta connected, and now that beta is connected to the gamma is connected to the methyls. So this is what is known as the leucine pattern. Now, as I keep on saying, yesterday I showed you the structures can perturb this a little bit chemical shift, but the pattern remains same. So that is where the problem comes with automation. So because the chemical shift perturbation ought to become difficulty, so what they do is they take the pattern of this, how the connectivity pattern, and then use it for automation. So then that's how this correlated spectra becomes useful. Now, as I said, that's the normal gradient COSY spectrum. We just worried about connectivities, so we can get it in 10 minutes or maximum half an hour. But if you want to get, now as you go to bigger and bigger peptide, there will be overlaps, or you want to remove the overlap, or you want to measure the coupling constant, then you have to do this COSY in a different fashion called the double quantum filter COSY. And that gives you a lot more resolution. Now, this is what is known as, so these are the whole connectivities, but what is more important is this NH to the alpha connectivity is called the fingerprint region. Now, you can literally count all that there are about 26 residues here. There are the 3 prolines. They won't give you this connectivity. So about 22, 23 connectivities are here. And now you see, if you simply blow this region, there are so many overlaps here. This would have been difficult in the gradient COSY spectrum. So that is why we have to do, to get more clarity, more time consuming DQFC. But if you look at the quality, is certainly much better, much useful, worth spending that time. Now, this COSY, the normal COSY would be a 2-pulse sequence, but here is one more pulse with a small microsecond time delay between them. Now, how it works and all, we have to go to the mathematics and see work out this trigonometric. This is simply an algebra, one can work it out. Now, in the textbooks, especially Cavino et al. Gives you every pulse sequences and their product operators and how it works and all. One can refer to that. Now, the advantage of DQFCs is both diagonal and cross peaks are absorptive peaks. They are quite sharp peaks. There are the dispersive peaks would be, the base would be broader. We will not go into those details because both are very sharp. You get very nice resolved resonances. The big disadvantage here is the signal to noise ratio goes down. So you need to give more time to acquire this data. So that was the proton-proton correlation. That's called the homonuclear COSY. Now one can get heteronuclear proton-carbon correlations as well. The CH correlation, CH carbon to proton, carbon to, in the methyl also C to that particular H. So this is the carbon-proton correlation. Now there is no diagonal. If you want a diagonal, it would be an enormous space, but you don't have enough space to store it, enough materials to resolve them. So what we see is only on one dimension, you have the proton chemical shift. In the other dimension, you have carbon chemical shift. What you see is only the cross peak. There is no diagonal here. Now, this also one can correlate. Once you have assigned a proton spectrum, that particular proton, if you go to the T1 dimension, that comes in the T2 dimension. And if you map it onto T1 dimension, you can pick up the corresponding carbons here. Now, in this particular case, I marked C beta, C gamma of the proline because it has a special case here. Because wherever there is a proline, it can occur in cis and trans. And the chemical shift of this C beta, C gamma carbon is important diagnostics for identifying whether it is in the cis form or the trans form. So one can do heteronuclear correlations also, and that's a different pulse scheme altogether. Now, likewise, one can do, when you consider peptides and proteins, the NHS plays an important role. So if there are some 36 residues, there will be 36 NHs. You can literally count the 36 NHs, and there are some side chain. Ignore this part. There will be 36 spots. So that way you can resolve it because you are expanding it 2 dimensions. So this is the proton dimension. This is the nitrogen dimension. That would be the pulse sequence. This is a more detailed pulse sequence called heteronuclear single quantum correlations. So now what you do here is, because the nitrogen has the magnetic moment is quite low, less sensitive. So what you do, you pull out the polarization with the proton, give it to the nitrogen. So all those things are done in this part, and eventually you detect it. And if you detect in the proton channel, it's time consuming. There are so many intricacies, we will not go into those details. But the same spectrum, you could have got it by simple two-pulse sequence. Because you're heteronuclear, you have to apply another pulse in the other nucleus also. So this would also given, but it would have consumed a lot of time, and there won't be this much clarity. So if you do all these kinds of gymnastics with these pulses, you will get a much better spectrum. Okay. Now, let us go through, because I would be taking a particular case of peptides. So I'm giving you, so there are these coupling patterns available. This is, I have taken from the FEG. I'm sorry, I forgot to give the reference here. But anyway, all these connectivity patterns. So these are the contour representation of the various connectivities of all 20 amino acids. Now, let us take the case of alanine. So alanine has an NH, and it has a C alpha and the methyl. So there are only 3 connections possible. So start with the NH, it couples to alpha coming around 4 ppm, it gets coupled to the beta. As I said, I'm taking only one side and ignoring the other side. So now this is the coupling pattern if you do a COSY experiment. Now, as I said, this pattern is very useful for automation purposes, even for manual assignment. So you can simply look at these patterns and pull out which residue is it in a complicated spectrum. Now, if you look at arginine, it looks so complicated, but it's no complicated. When I show you the real spectrum, you will understand that. Now, there is a series of connectivities from NH to the alpha, to the beta, to the gamma, to the delta. And there is another edge in the side chain, this is called the side chain NH, and you can retrace back. Now, from backbone, this is called the backbone NH, from backbone NH to the alpha, to the betas, to the gamma, and then to the delta. Now, this delta goes to the side chain NH. Now, you can trace it backwards also, from the side chain NH to the delta, to the gamma, to the beta, to the alpha, and to the NH. So there's advantage here. Though it's complicated, when you look at the 2D spectrum, it looks quite trivial. Now, likewise, every amino acid has a specific pattern. I'm not going into those details. I have anyway, whatever it is, I've listed all 20 amino acids, and each one has different pattern. Glycine is the simplest one. So there is this NH and this, the glycine CH2s are separated out. So there's, you get 2 peaks here, and this alpha are also connected. So this is histidine, leucine, isoleucine. So all 20 amino acids are listed here. Let us skip all those things. Now again, to once again repeat the same thing here. Now let us try to analyze. I told you in the beginning, only I picked up valine and leucine in the previous case. Let us pick up all of them now. Now, I've already given a proline assignments here, the proline 4 assignments here. Now, this proline is a special proline called the beta proline. One of the protons in the beta position is substituted by a methyl. So now again, I'm giving you only the partial spectrum from alpha to the rest of the connectivities. From alpha, now this is the alpha, go to the beta. Now beta is a methyl connection, so you get this methyl connection. Now from beta, it goes to the gamma, from gamma to the delta. Delta is again because it's cycles around and comes near nitrogen. Because it's nitrogen connected, it comes down. So alpha to the beta, beta to the beta, beta to the gamma, gamma to the delta. So that's how you pick up this residue. Now, likewise, already I have shown you how to pick up valine, alpha to the beta, beta to the methyl. So leucine also I've already told you, alpha to the beta, beta to the gamma, gamma to the delta and all. Now, this is a special residue called beta glycine. So, there are 2 alphas, 2 betas. You can pick that up also. And now there are 2 phenylalanines, phenylalanine 1, phenylalanine 2. It simply has this alpha beta connection. It won't have connection to the aromatic, which comes very much down. So but there are two, you cannot say which is phenylalanine 1, which is phenylalanine 2 from this, but then you have to do a combination COSY and NOESY spectrum. Then you can put the sequence, whether it is 1 or whether it's 5. So sequential connectivity comes from combination of COSY and NOESY spectrum. So we will see that a little later. So that's how we analyze a 2D spectrum, COSY spectrum. Now, as this keeps on size increasing, there is another version of this COSY called the total correlation time. It's also called HOHAHA experiment because two people developed it in different ways. Now, the main thing here is instead of second pulse, there is a long pulse here. It's called the isotropic spin mixing pulse. It can be a one long pulse here, or it could be made up of a number of 180-degree pulses, or it can be made up of all kinds of pulses, 90Y, 180X, 90 minus Y, and all, or another called DIPSY sequence. As I yesterday told you, all kinds of pulses are used here. 320, 410, 290, 285, and all. Now, if you use that kind of pulses, you get much better improved quality of the spectrum. So basically, it's simply a long isotropic mixing pulse. Now, what is the advantage here is, now earlier you go step by step, like going through the steps, but here it's like going through the ladder, one above the other. I'll show you in the next spectrum. So that's how it simplifies your analysis, because if a complicated spectrum, total correlation would be easier. Okay. Now in an amino acid sequence, it's simply made of amino acids connected by this peptide bond. And because of this peptide bond, the connectivity between the amino acids is lost. So now this is one amino acid. There are this alpha, beta, gamma connectivities. Now the blue one is one kind of spin system. The red one is another kind of spin system. The gray one is another kind of spin system. So all these things come up in one ladder. These things come up in another ladder. This separates out. So you can separate out the spin system, not in the stepwise manner, going one above the other, which I'll show you shortly. Now there are three sets of peaks showing different colors will be correlated. Now, any set of protons, unbroken J, so it has to be unbroken J coupling. So now this is coupled to this, this is coupled to this, this is coupled to that. But when you come this side, because this is 4 bond away, there is a connection breakage here. So all these things are connected, but this stands isolated. There is no connectivity at all. Now, what do you mean by isotropic mixing? It's simply a sequence of pulses that are designed to remove the chemical shape differences and create what is known as strong coupling environment. Essentially, in the long pulse, it takes it into a strong coupling environment so that all connectivities are linked to one another in a known set of a peak with unbroken J connectivity. Now, this is how the spectrum would look like. Now, this is the valence spectrum I have taken. Now, this would be the diagonal. So starting with NH, it goes to the alpha, alpha to the beta, beta to the methyl. So it goes stepwise. So the same thing can be clearly seen here. So this already I've shown you, NH to the alpha, alpha to the beta, beta to the 2 gammas, 2 methyls. Now in a TOCSY spectrum, you not only get the steps, you also get what is known as one above the other. NH is not only connected to the alpha, it's also connected to the beta, it's also connected to both the methyl. So you simply look at the NH peak, you get all the connections. So it's called the total correlations. So you need to look at only this region. You can forget about the rest of the region. Only NH region, if you look at, you get all the connectivities. So because if it is 40 residue, 50 residue, 100 residue, looking at the whole spectrum becomes difficult. But if you look at only this region, you have all this connectivity information. Now there are certain times how to use this, though those details are given here. Now, one drawback of this is there is another spectrum called rosy spectrum. So you may get some artifacts of that. And that is why they developed the DIPSY sequence, other sequences I showed you to clean it up. So depending on what kind of information you want, you can choose those pulse sequences and then do. Now this is a real example of, again, 1, 2, 3, 4, 5, 6, again, hexapeptide here. Now this is the TOCSY spectrum of that. Now, we don't have to worry about NH to the alpha, alpha to the beta and all. We simply look at the NH region. So if you simply look at that region, I've blown it up here. And now you can go one above the other in like a fashion and assign these residues. As I said, the resonance assignment is most important, and this is one of the steps in that direction. Now, how do we assign this here? Now, if you look at this, as I was telling you, arginine, though looks complicated, it's straightforward. Now, from NH, you have this alpha connection, and there is this beta connection, and then there is this gamma connection, and there is this delta connection. So NH to the alpha, alpha to the betas goes here, then gamma goes there, and then delta comes down. Now, the same way, you see another line going down there. This is from the side chain NH. Now, from the side chain NH, it goes to the delta and then to the gamma and to the beta. Now you see two parallel lines running. So if you see such a pattern, you can immediately think this would be the arginine residue. So though it's complicated, the pattern looks very simple. So you can simply pull out, oh, there is arginine present here. There could be more than one arginine present in a sequence, then there will be two sets of such things. So likewise here. Now there are two leucines here. Now leucine again has its own specific pattern, NH to the alpha, to the betas. There are again two betas and gamma. All of them come in a small range, and that is the two methods there. Now, because there are two leucines, there are two different correlations here, but we don't know which is leucine one, which is the other leucine. So either the leucine 1 or the leucine 5, we don't know. So again, we have to go to the NOESY to identify the sequence. But there are two leucines one can pick up. Likewise, one can pick up isoleucine, again, next to the alpha, alpha to the beta, beta to the gamma 1, gamma 2, and then gamma methyl and the delta methyl. So there would be 5 peaks around 220 ppm. So alpha is well separated out. Now this has its own unique pattern. So each residue has a unique pattern here. You can pull it out. Now the next is that there are two tryptophans. Again, you see those two tryptophans here, alpha to the betas. In one case, the betas are well separated. In another case, betas are not separated, but the pattern remains same. So this is how the TOCSY helps in assigning these residue-specific, what is known as residue-specific assignment, simply taking this region, looking at this region. You don't have to worry about the other region. Only case is proline. Proline won't have NH, so you have to go to the other region. Otherwise, most of the residues, you can pull it out from here. Okay, these are some more examples. So this is the corresponding 1D, and this is another cyclic peptide, and it simply traces of the NH alpha connectivity. Now this is the glycine residue, and this is the alanine residue. You see alanine has a different pattern, NH to alpha to the methyl, and that's the valine, and that is the leucine. And now there is one more there. Now this is called hydroxy acid, so let us not go into those things. I have not mentioned it here. So because that there are no NH, you cannot pull it out from here. You have to go to this region to pull it out. Now, okay, now all 20 amino acids, the patterns, I have put it in this cartoon taken from some textbook. So each one has a unique pattern. That's how it helps in automation and for also our manual assignment. I'll simply pick up some 4 or 5 to again represent this pattern. So this is the NH. Now these chemical shifts are unique to these things, but it could be small perturbation, but the pattern remains same. So that is the alanine pattern, that is the valine pattern, that is the glycine pattern, and that is the threonine pattern. Likewise, every amino acid has its own individual pattern, and looking, this is kind of useful for your analysis. Okay, so now that is about the first 2D experiment called the correlation spectrum. Let us move on to the other important factor or parameter called the NOESY spectrum. NOESY gives you NOE connectivity. So far in the COSY, they are all J correlated. information has to go through bonds, whereas in NOE, information goes through space. So now the NOE simple pulse sequence here, there are there are actually 3 pulses. And the mixing pulse has 290-degree pulses separated by constant time. Now the T1 is the regular, this is the preparation period, this is the evolution period. This keeps on varying between experiments, whereas these two, time difference between these two remains constant throughout the experiment. Now, what happens here is the first 290-degree pulses separated by increment T1. This time interval keeps on increasing between series of experiments. Now, what does it do? So this first 90-degree pulse flips into the XY plane. And now during this time interval T1, they start spreading out. They evolve according to the chemical shapes and the couplings. And now because they evolve, they rotate at different speeds. Now because they're rotate at different speeds, each one is identified. They're called flagged. Each one can be identified, given a number who is identified in this period because they rotate differently. Now, once they are identified, with the second 90-degree pulse, they are all put back to the Z direction. So that is where this phases comes. If you apply from X, it could be minus X, and these are all the details. Now, once they are identified, all are flagged, all are identified who is who, and they are all put back to 90-degree, put back to Z direction in the longitudinal axis, in the Z axis, then they are allowed to mix during this time. Now, this time, they exchange this magnetization between the nearby nuclei. And that is how they alter the population in the in the longitudinal plane. That's how it happens. Once that exchange takes place, you eventually detect and accumulate your data. Now, so the last two pulses separated by the fixed time, where the magnetization transfer takes place, resulting in a NOA buildup, which detected in the last pulse. Now, the only thing is there is a lot of intensity variation, which has a direct bearing on the distances and the quality of the spectrum. So it's better to give a very high resolution spectrum. You spend much more time in a NOESY data collection rather than the COSY data collection, because you don't want to miss any peak here. Every peak here has some information. Even in any minute peak would carry some information that would add quality to your spectrum. Now, the one disadvantage of this NOESY is its correlation time dependent, in the sense it's size dependent. Now, for small molecules, it will give a positive NOE. NOE, I told you, when you perturb certain nuclei, the intensity is perturbed on the neighboring nuclei. So in small molecule, the intensity increases, whereas in large molecule, the intensity decreases. But whereas in between size molecule like octapeptide, decapeptide and all, 1,000 or 2,000 molecular weight, now the NOE intensity goes to 0, so you lose this information. Now, the x-axis here is omega tau c. Omega being a spectrometer frequency, because it remains constant, we can simply worry about the TOCSY, the size of the molecule. When omega tau c becomes nearly equal to 1, then you lose that information. Now, there are methods to overcome that. We will see that as we go along. Now, the simplest method is simply record at a different spectrometer frequency, then this 0 gets shifted either downwards or upwards. Or you heat it up, the correlation time changes, you can also get it. These are the simple solutions. But there is yet another solution by the pulse sequence called the rho-Z spectrum. We will come to that next. Now, this is again another very useful, important. Now we have got the residue-specific assignments from the COSY spectrum. Now you can get the sequence-specific assignment because it can pull up connectivities from first residue, the second residue, the third residue, and all. So also it can pull up distance connectivities. If this protein fold and come nearby, as I said, from the first residue to the 100th residue come nearby, it can pick up that connectivity also. So the more the number of connectivity pick up, better would be the quality of spectrum. So, so the other way of collecting this data, whenever this omega tau c becomes 1, is collecting it by what is known as rotating frame experiment. So rotating frame overhauls or effect. Now, in this case, you get the same information, same NOESY information, but you overcome that 0 NOE region. Now, what you do is, instead of two pulses with a fixed time, here you use a long pulse. Now, this is very similar to the TOCSY spectrum I showed, but the power level and the time intervals are different here. So one has to be extra careful when doing ROSY experiments. Given a choice, you would do the NOESY experiment because it's a clean experiment. The ROSY experiments would have the TOCSY artifacts. One has to be careful when you do this experiment. But again, there are so many developments here, different pulse sequences develop, and one can go and take the help. But given a choice, I would do a NOESY experiment, a ROSY experiment, because it would have the TOCSY artifacts. Now, what is the difference here is, again, after the T1 spin, after labeling all these spin systems, you don't put it back to Z direction like in the NOESY experiment. What you do is, you hold it in the XY plane. You spin lock. That is why it's called spin lock. It's locked on the XY plane. Now it's independent of the main magnetic field. Now, magnetic field seen here is this B1 magnetic field, which is very tiny. Magnetic field equivalent to produced by this RF pulse. So then that is very, very tiny. But because of this tiny pulse, it's again like a strong coupling effect. So the magnetic field seen is very tiny. The omega dependent is very much less than 1. So it always gives you positive NOE. Hence, ROSY is always positive. There is no negative NOE here. It's all the time positive. Now, it is given by here. Now, these were the NOESY things which I showed you earlier. For small molecules, NOESY would give you positive. For intermediate molecules, it would give 0. And for large molecules, it give a negative NOE. When you talk of ROSY, it's all the time positive. So you are sure to get, so that there is no 0 region here. It's all the time positive. Now, one can, as I said, overcome this NOESY spectrum by going to different spectrometers, because different sizes goes to 0 at different levels. So one has to better have some idea of this correlation time, then you can pick and choose your experiments. Now, here are some standard thumb rules. If you want to collect a correlation time, take the molecular weight, multiply -12, you get an idea. And then you know where to pick it up, whether to do NOESY or ROSY, which spectrometer to use and all. Okay, now this is a typical NOESY spectrum to explain the difficulties in the peptides. Now, if there is a short 10-12 residue peptide, there is a chance you would miss certain peaks. Now you see in the NOESY spectrum, there are certain peaks missing, whereas there are a lot more peaks in the ROSY spectrum because all are positive here now. So there are other regions where you miss those peaks. Even here you miss some peaks. So that way ROSY is a useful for molecular weight around 1,000 to 2,000 things. So you won't lose any peak. And losing a peak in NOESY spectrum is very disastrous. So you should get as many peaks as possible to go to a structure determination. And now, here is another NOESY spectrum, whether NOESY and NOESY information content is same. Now it can not only give you the sequence assignment, it can also give you the secondary structure information from this NOESY spectrum. Now, when I say secondary structure, either it's a helical structure, I'm sure people are aware. When you talk of protein structures, it's made up of helices and extended sheets. Now, the helices has a certain pattern. Now, when you consider a helice, each amino acid NH is the adjacent, the distance between the NH of adjacent amino acids is quite small. It's called DNN, DNI to I+1, represented as DNN. So they become quite short. So 1 to 2, 2 to 3, 3 to 4, 4 to 5, 5 to 6, they're all are short distances, and it can be easily picked up by this NOESY. Now because this is inter-residue, there won't be J coupling because NOESY can pick it up because it comes nearby. So it's distance dependent. It can transfer magnetization from this NH to the adjacent NH. and this NH to the next adjacent NH. So likewise, you can pick up the sequence connectivity here. Now, the sequence connectivity combined with the correlation, J correlation connectivity, you can assign every peak in the given spectrum. Now, to stress on this, how this sequence connectivity helps in secondary structure assignment, as I said, in the helical cases, all these NH come nearby. Now you see a lot more cross peaks in the NH region. So we can simply concentrate on this NH region and blow it up. You can literally walk through this helix. Now, this is the particular sequence 18 residue peptide. Because the N-terminal attached to the Bock residue, you know the first residue comes, the highly shielded comes highest among the NHs. Now, if you pick it up, from there, you see a connectivity to the second residue, which happens to be AAB residue. That would be a singlet. In all other cases, all amino acids gives doublets. No, AAB is an unnatural amino acid, and it won't have the C alpha coupling to be a singlet. So you can pick that also. Now this, you can literally walk through the sequence 1 to 2, and this 2 is connected to the 3. So now 3 is connected to the 4. Now this is the 2-3 connection. That is a 3-4 connection. Likewise, 4-5 connection also one can pick up. So likewise, you can walk through. So 4 to 5, 5 to 6, 6 to 7, 7 to 8, and literally you can walk through this helical structure. So this not only gives the secondary structure information, it also gives the sequence information. Okay, now one more information, as I said, is not only the helical structure, one can also pick up the sheet structure. Now in the sheet structure, you won't see this NN and OE because they are far away. Now the NN goes far away, whereas the alpha and alpha of ith residue and the Y+1 residue come close by. Now this you can pick it up and build up this sheet connection, which is shown in the next diagram. Now, if you go back to this diagram, now this is the NOESY spectrum of sheet structure. Now you concentrate on this fingerprint region. There are very few NH-NH connectivity. Only whatever there is, it's only the turn connection or the long range connection. Now, if you blow this up, you can literally walk through this sequence. Now, blow this up. Now, this is 1 to 2 connection. 2 to 2 becomes very small, and then 2 to 3, 3 to 4, 4 to 5. You can literally walk through strand by strand. So that is how you get the secondary structure information, also the sequence information. So with that, I have covered the basics, 2 basic NMR experiments, COSY and NOESY. Also I've covered HSQC, heteronuclear correlation experiment. There are many more experiments one can do. Again, varieties of COSY experiments, varieties of NOESY experiments, varieties of HSQC experiments. And if you go to 3, there are a lot more experiments we do. Now, if you have these basic experiments, one can get now go ahead and get the structures. Now, this is the protocol for structure determination. You should now the first thing is you should have a good 1D spectrum, or the HSQC provides a good 2D starting spectrum. Now, once you have this, as I told you, if it is not good spectrum, you have to do something, change the solvent, change the temperature, do something else, get a decent spectrum, then only you can proceed to do all of the time-consuming experiments. Otherwise, you will be wasting your time. Now, once you have got a established good monomeric spectrum, it could be aggregations also, so one should take care of those things. So once you have a decent spectrum, then you do these homonuclear, heteronuclear experiments like TOCSY, NOESY, which I discussed so far, to make this resonance assignment. Now, the resonance assignment is the most time-consuming. You cannot go wrong here. It should be a foolproof. So you can do more experiments. You can overkill the experiment. Just get the same information by different 2D experiments, but you have to be sure. You can never go wrong here in this step. So if it is ambiguous, leave it out. Only very unambiguous things, you pick it up and then come back and look at it again, and then you will improve your data. So you have to use the combination of this TOCSY, COSY, NOESY, HSQCs, and all to get this. And as I said, this is the most time consuming. You spend a lot of time here. Now, once you have done the assignment, the rest are all straightforward. Now, you have to do the secondary structure assignment using the NOESY, as I told you. And NOESY provides you a lot more peaks. So we will come to that later. And then the J coupling values, and one can get the hydrogen bonds through the chemical sheet, which I did not explain. But various temperatures, if you do, you can break the hydrogen bonds and estimate which are the amides which are hydrogen bonded, which are not hydrogen bonded. That information you can get from J coupling. You can get the dihedral angle information. And through sequence, you can get the backbone information. And once you have established, you have a fair idea of secondary structure, go to the long-range NOEs. Now, as I said, it's not only the sequential, you can get 1 to the 5th residue, 5th to the 100th residue, 2nd to the 10th residue, or 4th to the 111th residue, and there are a lot more NOEs because this structure twists and turns. You can pull it out, all those things, and list it out. So that's where the NOESY and ROSY becomes very important. So this is the main backbone of the structure determination so far. But eventually, people want to get it straight away from the chemical shift. That's a long way ahead. But at the moment, still depend on this NOESY and the ROSY. So this is the basic experiment. Now, once you have done that, you take all this information, and there are structure calculating program available on the computers. You use all these NMR parameters as constraint, the distance constraint, mainly distance obtained. All these intensities of the NOESY peak can be converted into distances, and the dihedral angle information is obtained from J values. Hydrogen bond information you obtain from the NH hydrogen bonds, and all these things, or even chemical sheet information, as I told you, from the chemical sheet index plots. All those things, even you have some secondary structure information. All these things, you feed it into the calculating program they use as constraint, and it will generate a structure. Now, here is a typical example. I'll go through this. So record a 1D spectrum. So it should be well resolved. Now, this is a hairpin structure, extended hairpin structure. Here, this is an octopeptide, simple peptide, and they're very well resolved. That's why I took this example. Now, do this TOCSY experiment. These are the partial regions. So you can assign residue by residue, which is valine, which is tryptophan. Now, valine 3, valine 8 is difficult to distinguish. You use the NOESY experiment and identify which is valine 3, which is valine 8. Likewise, this is simply the NH region. So all the residues can be picked up here. With the NOESY connectivity, the sequence connection also can be picked up. If there is a proline, you won't get it here. You can get the connection from the rest of the region. Now, and some automatic information also you can get. And once you have this information, you go to the NOESY spectrum and get, there is a sequence information also. There is long-range information also. Now, there is this automatic region. Because it projects out, it gets connected to the various protons around. And there is a lot of long range. You see the Y2 alpha gets connected to the Y7 alpha. So this is the second residue, the 7th residue. Because it folds around, it comes nearby, and the NOE can pick it up. Likewise, this L1NH and V8NH can be picked up. Now, this is the peak corresponding to that 1 to 8 NH. Likewise, you can connect 5 to 6. And this is, of course, sequential 2 to 8. 2 to 8 connection is picked up here, so that likewise you pick up every possible connections. And now once you look at this, these intensities differ. If they are close by, you get bigger intensities. If it is far away, less than 5 angstrom, you get smaller intensities and all. And this is the hydrogen bond information. And once you get all this information, you can convert these intensities to the distances. So you can crudely consider as if there's a very strong peak, you can constrain your distance. The lower limit is always Van der Waals distance. It always remains 1.8. It's only the upper distance which we have to worry. So if it is a large NOE, you constrain it less than 2.2. If it is small NOE, you constrain it to the maximum value possible, that is 5 angstrom. If it is in between, you constrain it to 3.5. This is the crude classification. One can classify into 10 different regions according to number of contours and all. but this is the minimum you can do. And you have other information, hydrogen bond information, dihedral bond information. You put all these things into a computer program called that Cyana. Cyana is one of the most useful software. And now once you feed all those things, you get a structure. Now, normally in NMR, you get a bunch of structures because you're not sure of these distances. You can only estimate these distances. As I said, and then you get a range of distance, but not a fixed distance. But because you get a range of distance, all of them satisfy this criteria, and you get a bunch of structures. Now you get more and more NOEs, the bunch clumped together very well. So the RMSD between these structures determine the quality of your structure. Normally, for this octopeptide, you get about 100 distances, about 6 dihedral angles in this particular case, 4 hydrogen bonds. This is at least for every residue, you should have some 10 distances, then your structure would be neat. So this is how you get a structure. Once you get a structure, you validate by what is known as Ramchandran map. I have not told you this, you may have to read it here. Now, he told you the whole protein structure is defined by the 5-side dihedral angles. And now this 5-side can take up only certain, this is the 5-side map, also known as the Ramchandran plot, is given by Professor Ramchandran. This is another story to tell you in some other time. And now you validate your structure by projecting the 5-side values obtained from this structure to this map. Now, there are only certain 5-side values allowed. Now, if they come within that range, then your structure is good. But if these 5-side values goes above the allowed region, then your structure is not good. You have to go back and refine your structure. Now, when you talk of refinement of structures, this is the NOESY and COSY are the basic things. There are other things called residual dipolar couplings and all. They are more involved. One can refine your structures and improve the quality of your structures. So now that is the thing. Now I have time. I'll go through one real case study. Now this is a study of conotoxins, a project undertaken by us in collaboration with Professor Balram and Professor Krishnan from NCTS. Now, he's a very good, Professor Krishnan is a great scientist. All these scientists are passionate for doing science. But he's no more. He expired a few years ago, and he's in the biology department. He was in biology department at CBS. Now, these conotoxins got important because now they are developed into drugs. Now, what are conotoxins? They are the toxins produced by these conus snails. You would have seen all these conus in the seashore. They are all living organisms, but when you collect them, they are all dead. Now, they produce certain peptides, which are very useful drug molecules. So that is why we took under this study. Now, there are all over the world, there are 500 species of these conus snails. In India, we have identified about 77 of them. And they are produced by snails called the conus. And the length varies about 8 to 50 amino acids, easy to understand. The main important criteria is they have this disulfide bond. Now, in India, you find these conus snails in the seashores, and Professor Krishnan has such a passion about this science. So he used to literally go to the seashore, interact with the fishermen, collect this conus snails, and either in the Tamil Nadu coast, Tuticorin, I think, or in the Mumbai coast, and come to the lab, extract these peptides from them, and then analyze them. Now, all these things started because there's another person called Professor Oliveira in University of Utah who done extensive research on this. He would be the person waiting in the Nobel Prize, and he has literally developed drug molecules of painkillers, very useful for cancer patients. They substitute the morphines and all. They can be substituted for such things. Now, this is a slide to tell you the curiosities of the students. Now, in his lab, there's a student who worked, they isolated so many peptides. Now, they had these animals also in their labs. Now, what he did was he simply took various peptides and introduced them to these rats in his lab, and each peptide has a unique property. So some has the head swinging, some has circular motion, some has kicking, some has depression. So this is kind of a combinatorial effect which is used by snails to get food from their prey. So now that's where it all started. Coming back to our, cutting the long story short, coming back to our analysis, now these corner snails, we concentrated on conotoxin, on a particular peptide called contrafans. I'll come to that in the next slide. Now, the pharmaceutical properties, it targets various ion channels. Most of them, this works on these ion channels, very potent and highly selective. Now, this is how the coners use this drug. They are all these toxins are stored in their body, and there is a cone coming out. Now, they are so slow moving. Now, when they see a fish going around, they're fast moving, they simply pump up this toxin and then paralyze this prey. And then that's how they get their food. Now, when you concentrate on this particular contrafan, one particular peptide, now they are, from pharmaceutical point of view, they are water soluble, they are low molecular weight and simple to manufacture. Now, we will take a case of this contrafan because this acts on the calcium channel, and the one specific reason is this drastically improved the current in the this calcium channel. Now, how do we go about it? Now, collect this crude venom and purify them by HPLC, and then use mass spectrometry to identify the various groups and all. Use UV spectroscopy and then identify the aromatics and all. and then by mass spectroscopy, you can get the sequence. So once you get the mass spectroscopy, NMR, all these combinations of the spectroscopy also, because these are small peptides, one can get the sequence. And now these are produced in so low quantities. Once you know the sequence, you can chemically synthesize them. Once you chemically synthesize them, you should know how the folding pattern and all can be identified by NMR. The electrophysiology is done to get this ion channel activity and all. The bioactivity is all done, and NMR especially is used to get these 3D structures. Now again, the same picture. Now we concentrate on this contrafan, called contrafan because it has tryptophan in it. Now, normally, it's about an octapeptide. Now in this case, we went through all those mass spectroscopy, other spectroscopic techniques, and eventually got this sequence and synthesized this molecule. And it becomes active only when this tryptophan is the D isomer. Normally, in any peptide sequence, there all be L isomer. In this particular case, this happens to be the D isomer. Now, this is called contrafan laurosii because it's obtained from that species. And this has a molecular weight of 959. So henceforth, we call it LO959. Here's the sequence. And why we picked up is this has unusual biological activity, increases the calcium current, contrast to the known contrafan functions. So you did all these NMR experiments and finally energy minimized this model to get the structure. Now, we had a surprise to begin with. So as we purified and all, when we run the HPLC, you see two peaks. It's a purified sample. Now, when we start worried and then cut off this first peak and then re-inject it, and you get two peaks. Cut off the other side, re-inject it, it's two peaks. So we took some time to understand this. This is because this sequence has a proline. It exists in two forms, cis form and the trans form. So there are literally two peptides here, two different structures simultaneously present that we came to know when doing NMR because all the resonance is doubled. So when we do, this is done on a 700 spectrometer with a cryoprobe, very good sensitivity. We can work with low amounts of samples and all. So now, again, to make sure that the tryptophan active, so that has a good clean spectrum, whereas the L-tryptophan didn't have a decent spectrum. So this is also the active moiety. So we took this up, and then this 1D NMR, now you see a clean 1D NMR spectrum. And then because it's complicated, I showed you this also in the very beginning of this lectures. And we need 2D experiments to do this. Now, this is HSQC spectrum just to show you everything is double. So there is a red and green component. So simply show there are two kinds of peaks here. Now we have to go back to the TOCSY spectrum, get the residue-specific assignment, which I explained to you, and again the proline, which cannot be obtained in this NH region. We have to go to the rest of the region and pull out the proline resonances. And we have to use the NOESY spectrum also to get the sequential assignments. And not only that, now the NOESY gives you all kinds of NOE, long range NOE, medium range NOE, and all. And now we use the HSQC spectrum, as I told you. The carbon spectrum identifies the cis trans isomers because the difference between the gamma and delta tells you the proline 2 happens to be the cis form, whereas the proline happens to be the trans form, because the difference here is large, difference here is small. So, and that's how we pick up two sets of things. So we independent, and this is the HSQC spectrum where you can differentiate between the cis and trans isomers. And now we go back and use all this NMR information, eventually get two different structures because there are the two forms. Why these different structures? These are called shape-shifting molecules. This is the efficient way and these snails use. The same molecule exists in two different forms. They can bind to two different receptors. So there's one efficient way of doing it, and we could publish this way back in. So this is how NMR is used to get these structures. So one can develop it further, and I'm not going to those details here. Now with that, I've come to the end of my lecture. And towards the end, I should acknowledge my mentors. Professor Ketrapal is the one who brought NMR to the India. The first NMR spectrum, 270 megahertz spectrometer came under his leadership. And he's bold enough to take that decision because helium was a big problem. So 4 institutes associated with it. And he took the lead and established that. And after that, as you see, there are nearly 600, 700 NMR instruments in this. Now he's an institution builder. He started our NMR center. Later on, he went on to the Vice Chancellor of Allahabad University. Later on, he started in Lucknow, what is known as CBM, currently known as CBM, Center for Biomedical Research and all. So, so we all started working from him. Now, then Professor Anil Kumar is another big person in NMR. He was a big professor. He's the one who knew NMR in those days. He was worked with both Nobel laureates, Ernst and Utrecht, and he holds the record of getting the first NOESY spectrum. That is where the protein structure started. And then Ketrapal picked him up. And when we started NMR Center, he was the first person who he taught us quite a bit. We learned a lot of academics, NMR principles from him. He's the one who pushed me to take up this NMR. Eventually, Professor Balram, who was the director of the institute, also has a great contribution in peptide NMR. He was also my research guide. I learned a lot from him, and also continue to collaborate with him. So not only them, there are in NMR Center, we have a lot of our colleagues, a lot of other associates in institute. From all of them, we learned. And so I take this opportunity to acknowledge all of them. Now, finally, I want to end up with this slide. Shows while doing science, you collect a lot of facts. You need a lot of facts, but you have to put it in a particular form. If you don't put it in a form, it's like building a house from stones. If you don't put it in proper form, it looks like a bunch of stones. They're no more science than a heap of stones. So if you put them in order, you get a nice house. So science is also like that. You simply collecting data, facts is not sufficient. You have to build a story around it, then only it looks nice. So with that, I end my lecture series, and I hope I have given you some exposure to the NMR And thank you all for your attention. Thank you. So with that, I end this lecture.
Thank you very much, sir. Thank you for all the deliberations which you made for these long 3 days. Really, when I was thinking that when I was just shaping this NMR lecture series, I thought that 3 days will be a big time, and it will be like cumbersome for you to just spare these 3 days for us. But I'm very happy that you very benevolently spared these 3 days for us, and you could just lead us through all the nuances of NMR, and we could hear from you, we could learn from you. Reading from books is one thing, but learning something from someone who has been into that process and who has been into that instrumentation process is really great. Because what we do, we learn from books only, but whatever you have told us, these all were the excerpt from your own experiences. So we are really thankful to that. Now I will invite principal of the college, Dr. Arpan Bhagwaj. He himself is a professor of organic chemistry. So he, with principal, he has that edge for chemistry too. So I invite you, sir. Please go ahead, sir. Dr. Ragothama is with us.
Okay, thank you, Ms. Dr. Kalpana Singh. It's really a great pleasure that the Ganga was flowing in the campus of the Madhav Science College, and students were just I don't know what should I say, but as Madam Singh knows, because of administrative responsibilities and admission is going on in the college, so I couldn't join all the time. But I go home, and in the evening I like to listen and see the things what happened today. And I was really I was more than happy, you see, because I must not say much about NMR before sir, because what he has taught us in last few days, it's really wonderful. Yesterday when he was talking about chemical shift and NOE and everything, it was really the way in which he was presenting the things that taught us that how should we go before our students and what should be our preparation. So almost every aspect of the spectroscopy, 1D NMR, 2D NMR, COSY NMR, and NOI, and everything, whatever he could do in a limited time, he managed to do all those things. And looking at our request, he made the things simpler, because in a place like Ujjain, we don't have students we want to make them of the caliber of IASC, and that we can do only through our faculty. So in that series only we have requested sir to deliver his things to our students, and it was really wonderful to listen to you, sir. So I thank you once again. And whenever the things become normal, this is a new normal time, but when the normal time will come, we will be very happy if you can come to Jan and can grace our college by your presence. So I once again thank you. And it was really as in your presentation, I just saw in the last that you were talking about Professor Anil Kumar, and he recorded the first spectrum of his own sort, which you were just discussing. So through you, we could come to know about him, and this is what the chain reaction is called. Like my words will be incomplete if I don't thank Dr. Srinivasulu, Madam Srinivasulu, and Dr. Srilakshmi, who could make this thing possible. So this is the real time to remember our teacher. Our teachers, not only teacher, of course, Dr. Srinivasulu, who his family has established this chair, and we could organize this lecture. We are organizing lectures through that chair periodically. And I thank once again to Dr. Prabhalu madam and Dr. Sri Lakshmi Lu also. And once again, I request you to, sir, hold our hand and our students to so that we can make things better. Thank you very much, sir. Thank you.
Thank you very much. Very nice of you. Thank you, sir.
Thank you, sir.
This lecture series was organized under the aegis of Professor K. Srinivasulu Chair, which has been established in this college by his family members. Dr. Prabha Srinivasulu, who herself is a esteemed scholar of history, and Dr. Sri Lakshmi Desiraju, who also happens to be alumni of this college, as well as daughter of Dr. K. Srinivasulu. as much as I know about, and I remember about Professor K. Srinivasulu as my guide and as a teacher of chemistry, Professor K. Srinivasulu himself was very much attracted towards instrumentation. He always talked about instruments and instrumentation, whatever subject you are doing, whatever you are learning, but he would just always mention about all these instruments, and he would just always mention about new technology. He would just say that you students should be more research, otherwise sticking to old things will not fetch you many new things in the research. So in this way, this lecture series again proved to be a boon and a very relevant one when we talk about the thing that this is organized under Professor K. Srinivasulu chair. Definitely, as the coordinator of Professor K. Srinivasulu chair, I would like to thank Dr. Ragothama, Dr. Arpan Bhardwaj as the principal, and Dr. Sri Lakshmi Desiraju for being the founder member of this chair and for being a very outstanding support. Now, I would like to request Head of the Department, Chemistry, Dr. Brajesh Pare, as Chemistry Department is one of the hosts of this program. Sir, please come and extend your official vote of thanks. Dr. Pare, floor is yours.
Unmute. Yeah, thank you. Thank you. I'm audible now?
Yes, sir,
Yeah.
you are audible.
Yeah, thank you, Professor Ragapati, for a wonderful talk. And this is, in fact, the first talk on after the chair in the name of our teacher, Professor Srinivasulu. And really, it's very sentimental and a very emotional moment for us. So thank you for, sir, all your academic support and the 3 days, somehow, because of this new normal, our students, really were not in a very coherent manner. So, but anyway, it was so wonderful, and next time, definitely, we would like to have more students to take part in it. And once we go into the normal situation, sir, please do come to our institute and deliver your talk on that. And it was really, really very enriching. I take this opportunity to recall our teacher, Professor K. Srinivasulu, and I'm thankful. I'm really thankful to Mrs. Lu, Dr. Shri Prabha Lu, and Dr. Shri Lakshmi for having spared. Just supporting Madhav Science College for establishing this prestigious chair. Because when recall my teacher, Professor Lu, he was a very, very dedicated researcher, a wonderful teacher, and all through his life, he was so keen in guiding the students, and he was really, he was very fond of spectroscopy. So what could be more, to have a first lecture in his memory, that too on spectroscopy. Thank you, Professor Agapati, and we are thankful on behalf of the Department of Chemistry, Madhav Science College, Ujjain, and keep guiding us and keep inspiring us. Thank you very much. Thank you.
Thank you, sir.
Thank you, sir. Thank you, Dr. Ragothama, for being with us for these long 3 days. And as Principal Sir has mentioned and HOD Chemistry has also mentioned, we would just love to have you physically with us. As you know that Ujjain is a city of Lord Mahakaleshwar. Mahakaleshwar is here, and Mahakaleshwar is one of the 12 Jyotirlings in India. So you would just Dakshin Mukhi bhi hai. So we will just love to visit this place if you have not visited earlier, I suppose. So please do visit.
Yeah, certainly I'll take. I'll certainly. I think I have come once in a quick visit, but now you invited. I surely come sometime. As your principal said, when things get normal, we will make a visit. I also take this opportunity to thank Sri Lakshmi, who made this connection possible.
Yes.
I think she is in the audience.
Sri Lakshmi, I cannot see because of certain, I think, engagements. If Sri Lakshmi is there, Sri Lakshmi can. She was here on first day, but I don't think so that she.
No problem. Okay.
Yes.
Yeah.
Okay.
So.
Thank you, sir. Thank you very much for being with us. Thank you, sir.
Okay. Yeah. So we close now.
Yeah, sir. Thank you, sir.
Okay. Thank you.
I have an announcement for students. As I told you, there is I will be uploading assignments at 8 p.m. and there will be a window period for 2 hours. I'll be closing the assignment at 10 p.m. Assignment is being given to us by Dr. Ragothama himself. So please do attempt the assignment. It's a graded assignment with 25% weightage for your comprehensive cumulative evaluation. Thank you very much. Thank you, students also. Thank you for being with us. Thank you, sir.
Thank you.
As this was an academic lecture series, we are not hosting any of the feedback link because academic lectures are not open for feedback. I do not believe in that, opening academic lectures for feedback, and that too of caliber of Dr. Raghuttama. So we are not in a position to give any feedback. We only have to answer the questions which will be uploaded in the form of assignments at 8 p.m. So please do check that. And for the students, I'll talk to you later on, because this is not an exact platform to talk about the issues which I want to talk, but I'll be talking either on Google Meet or on the Google Classroom, which I have. So we'll be talking there, because your attempt this time has many questions, and these questions has to be solved. There are many question marks, and these have to be solved before we proceed with the regular classes on spectroscopy. At least I'm not talking about other subjects, but before we take up classes with spectroscopy, we need to answer, actually you need to answer me certain things. Only then I will be taking up your further classes. Okay, thank you very much. Thank you for being with us. Mr. Administrator, you can close the meeting now. Thank you.